Hello everyone, welcome to the channel Easy Life Science. In today's lecture, we will learn about how we going to derive energy from food we ate. What are all process takes place in the body to convert various kinds food into the energy required for the body. Biological oxidation is the process that takes place within the living entities to yield the energy required for the body through the degradation of intakes in the form of food. Caloric value of food. Usually, we measure energy in terms of calorie, kcal. Then what is calorie? Calorie is the amount of energy needed to raise the temperature of 1 kilogram of water by 1 degree Celsius. The energy accumulated in food substances in the form of proteins, fat, and carbohydrates, the energy value of foodstuffs, expressed in calories or kilocalories. The caloric value is determined by the presence of unoxidized atoms of carbon and hydrogen. A molecule of fat contains more unoxidized atoms of carbon and hydrogen than a carbohydrate molecule or a molecule of protein. That's why fat yields more energy than any other kinds. 1 g of fat yields 9.3 kilocalories, 1 g of carbohydrate 4.1 kilocalories, and 1 g of protein minus 4.1 kilocalories. Although fat yielding, but why it is unhealthy? Why only sugar molecules are considered as easy way energy molecules? We will learn that in detail. The synthesis or degradation takes place within the cells called as cellular metabolism. Cellular metabolism. All of the chemical reactions that take place inside of a cell are collectively called the cell's metabolism. Cellular metabolism may be catabolism and anabolism. Catabolism breaking down of large complex molecules. Example, respiration, glucose plus oxygen gives carbon dioxide plus water. Hydrolysis, starch and DNA, where it gets breakdown into individual moieties such as glucose and mononucleotides. And catabolism reactions are usually exothermic in nature. Anabolism is synthesis of large complex molecules from small molecules. Example, protein synthesis, polymerization. And polymerization of mononucleotides into DNA. These kind of reactions are usually endergonic, take in energy. Then how body will get energy through these reactions? ATP, adenosine triphosphate, composed of base adenine, ribose sugar and 3 molecule of phosphate. It is called universal energy currency because energy is usually stored in molecules of ATP. Whenever something needs energy within the body, usually ATP undergo breakdown into ADP, which results in release of inorganic phosphate. Breakdown of any food molecules in the body will take place in 3 stages from eating ultimately lead to energy production. Stage 1. Stage 1 begins when we eat food. It gets mixed with several hydrolysis enzymes, such as starch degrades into sugar by amylase and amylopectinase enzymes. Lipids by lipases. Proteins by protease, etc. Stage 2. Stage 2 usually results in ATP production. When glucose formed in stage 1 undergo series of reaction called as glycolysis. Lipids through oxidation yield several molecule of ATPase. Stage 3. Stage 3 occurs in mitochondria in the cell, which is mainly to electron transport chain. Okay, now we will learn about how carbohydrates, that is glucose, yields energy to the body. Once starch or any polysaccharides break down into simple sugar molecule glucose, it undergoes series of enzymatic reactions through the process called as glycolysis. Glycolysis. Glycolysis is glucose catabolic pathway. Glycolysis pathway comprises of total 10 steps, and those are broadly divided into two phases. 1. Preparative phase, step 1 to 5. 2. Payoff phase, step 6 to 10. Preparative phase. This is the first phase of glycolysis. In this, 6-carbon glucose gets converted into 3-carbon molecule glyceraldehyde, 3-phosphate and DHAP, dihydroxyacetone phosphate. This phase contains 5 steps. Step 1. Phosphorylation of glucose in presence of enzyme hexokinase, a phosphate group from ATP added to glucose and forms glucose 6-phosphate. Step 2. Isomerization. Glucose 6-phosphate gets converted into fructose 6-phosphate by the enzyme phosphoglucoisomerase. Step 3. Phosphorylation. In stage 3, one more phosphate from ATP gets added to previous molecule, results in fructose 1,6-bisphosphate. Step 4. Condensation. This is the major step in glycolysis where 6C molecule breaks apart to 3 carbon molecules, namely dihydroxyacetone phosphate and glyceraldehyde phosphate. SWIT P5 isomerization. The glyceraldehyde 3-phosphate enters into the next step, whereas the HAP gets converted into glyceraldehyde 3-phosphate by the enzyme isomerase. At the end of the preparative phase, 2 ATP molecules gets consumed and 2 3-carbon molecules were formed. 3-carbon molecules enters into the next phase called as payoff phase. This is the second phase of glycolysis. In this phase, glyceraldehyde 3-phosphate is converted into pyruvate molecules. This phase have 5 steps. Step 6. Dehydrogenation, where NAD+ gets attached by hydrogen by the enzyme hydrogenase and forms NAD, and also triose phosphate dehydrogenase adds to phosphate, results in 1,3-bisphosphoglycerate. Step 7. Substrate level phosphorylation. Step 7 is first step of ATP yielding stage in glycolysis. 2 molecules of ATP gets produced by taking phosphate from the previous 2 molecules of 1,3-bisphosphoglycerate with the help of an enzyme phosphoglycerokinase. Step 8. Dehydration, which is intramolecular rearrangement. 2 molecules of 3-phosphoglycerate in presence of enzyme phosphoglyceromutase results in formation of 2 molecules of 2-phosphoglycerate. Step 9. Enolization. 2 molecules of 2-phosphoglycerate with enolase yields 2 molecules of phosphenol pyruvic acid, PEP. Step 10. The last step of glycolysis is substrate level phosphorylation. 2 molecules of PEP plus pyruvate kinase gets converted into pyruvate by the enzyme pyruvate kinase, and this step also yields 2 ATP molecules. Stoichiometric equation of glycolysis. 1 glucose plus 2 ATP with enzymes in 10 steps ultimately yields in 2 pyruvate plus 4 ATP plus 2 NAD plus H plus H2O. So that the total net yield of a glycolysis cycle is 2 direct ATP yield and 2 NAD molecules. 1 NAD molecule is equivalent to 3 ATP, so that direct ATP yield per glycolysis is 8 ATP molecules. Krebs cycle. Once pyruvate gets produced in glycolysis, it's entered into next stage or simply cycle, which is called as Krebs cycle. The series of reactions in the cycle was identified by a British scientist, Hans Krebs, and hence the name. The citric acid cycle, also known as the Krebs cycle or tricarboxylic acid (TCA) cycle, is the second stage of cellular respiration. The first phase of cellular respiration, called glycolysis, takes place in the cytosol of the cell's cytoplasm. The citric acid cycle, however, occurs in the matrix of cell mitochondria. The pyruvate from glycolysis become acetyl-CoA by the enzyme dehydrogenase in the Krebs cycle. This acetyl-CoA, which forms citric acid by the enzyme citric synthetase. This citric acid is the first stable product in this cycle. Hence, this cycle is also called as citric acid cycle or tricarboxylic acid cycle, TCA cycle, which later undergo series of enzymatic reactions to yield ATPs. In eukaryotic cells, the citric acid cycle uses one molecule of acetyl-CoA to generate 1 ATP, 3 NADH, 1 FADH2 to CO2, and 3 H+. Since two acetyl-CoA molecules are generated from the two pyruvic acid molecules produced in glycolysis, the total number of these molecules yielded in the citric acid cycle is doubled to 2 ATP, 6 NADH, 2 FADH2, 4 CO2, and 6 H+. The NADH and FADH2 molecules produced in the citric acid cycle are passed along to the final phase of cellular respiration called the electron transport chain. Here NADH and FADH2 undergo oxidative phosphorylation to generate more ATP. The another process of glucose breakdown is fermentation. Fermentation is the anaerobic process of conversion of glucose into alcohol. There are mainly two fermentation pathways, lactic acid fermentation and alcoholic fermentation. Both of these pathways begin with pyruvate, which was produced as the end product of glycolysis, are anaerobic, require no oxygen, and allow glycolysis to proceed aerobically. A relatively small amount of ATP is generated in the pathway, but that's about the best that can be accomplished in the absence of oxygen and mitochondrial respiratory chain reactions. Lactic acid fermentation, cheese, and yogurt making anaerobic muscle activity. Pyruvic acid gets converted into lactic acid with regeneration of 1 mole of NAD+ per pyruvate or 2 moles of NAD+ per glucose molecule. This conversion is catalyzed by lactic acid dehydrogenase. Alcoholic fermentation, beer and wine making. Pyruvic acid in absence of oxygen gets converted into ethanol with release of carbon dioxide, with regeneration of 1 molecule of NAD+ per pyruvate or 2 moles of NAD+ per molecule of glucose. Alcoholic fermentation is a two-step process, catalyzed by pyruvate decarboxylase, forming acetaldehyde plus CO2, and alcohol dehydrogenase, forming ethanol. Thank you for watching. In the next video, we will learn about the biological oxidation of proteins and lipids to yield energy.